A recent study published in Cellular Signalling by researchers at Osaka University has identified a novel molecular mechanism underlying the morphological changes in senescent cells, proposing the protein AP2A1 as a potential biomarker and therapeutic target for age-related diseases. The aging of organisms is associated with the accumulation of senescent cells, cells that have ceased to divide and exhibit significant alterations, including a notable increase in size. This study investigated the molecular mechanisms driving these changes, focusing on the protein AP2A1, alpha-1 adaptin subunit of the adapter protein 2. The findings show that AP2A1 is upregulated in senescent cells and localizes along actin stress fibers, which appear thickened and more stable. The reduction of AP2A1, knockdown, in senescent cells induced a rejuvenation phenotype, restoring smaller cell size and proliferative capacity. Conversely, its overexpression in young cells accelerated the onset of senescence markers. The proposed mechanism suggests that AP2A1 facilitates the transport of integrin V1 along stress fibers to reinforce focal adhesions, thereby enabling the cell to maintain its large architecture. These results, also validated in UV and drug-induced senescence models, indicate that AP2A1 is a crucial modulator of the senescent state. Introduction to cellular senescence. Cellular senescence is a biological process wherein cells enter a state of permanent growth arrest. This phenomenon is a response to various stressors, such as telomere shortening during cell division, replicative senescence. Senescent cells accumulate in tissues with advancing age, contributing to numerous pathologies, including neurodegenerative and cardiovascular diseases, as well as cancer. One of the most conspicuous features of senescence is a drastic increase in cell size and a flattened morphology. These changes are closely linked to a profound reorganization of the cytoskeleton, particularly of stress fibers, which are bundles of actin and myosin responsible for contractility and cell adhesion. Despite these observations, the molecular mechanisms governing the maintenance of this imposing senescent cellular architecture have remained largely unknown. Building on a previous proteomic study that identified 63 upregulated proteins in the stress fibers of senescent human fibroblasts, this research focused on AP2A1, a protein known for its role in clathrin-mediated endocytosis, CME, whose connection to senescence had not been previously described. Study findings. Characterization of senescence and the role of AP2A1. The researchers utilized human fibroblasts, HFF1, brought to replicative senescence through serial passaging in culture, up to passage 30 P30. The aged cells, P30, displayed the classic hallmarks of senescence. Increased size. The cell area increased 6.6-fold compared to young cells, P10. Senescence markers. They exhibited increased activity of the enzyme SABU-GAL and higher levels of the proteins P53 and P21, known cell cycle inhibitors. Altered stress fibers. Stress fibers were significantly thicker, more stable, and had a reduced turnover in senescent cells. The analysis confirmed that the expression of the AP2A1 protein was significantly elevated both in the total cell lysate and specifically in the stress fiber fraction extracted from senescent cells. Super-resolution imaging revealed that AP2A1 localizes along the surface of individual stress fibers, suggesting a close interaction. AP2A1 as a modulator of senescence. To determine whether AP2A1 was a cause or a consequence of senescence, the team conducted knockdown, gene silencing, and overexpression experiments. AP2A1 knockdown, rejuvenation. By silencing AP2A1 in senescent cells, P30, the researchers observed a reversal of the senescent phenotype. The cells became smaller, stress fibers thinned, levels of senescence markers, P53, P21, SAB-GAL, decreased, and most importantly, the cells regained their ability to proliferate. This phenomenon was described as a genuine cellular rejuvenation. AP2A1 overexpression, induction of senescence. Conversely, when AP2A1 was overexpressed in young cells, P10, they began to exhibit characteristics typical of senescence. They increased in size, displayed thickened stress fibers, and elevated levels of P53 and P21, while their proliferation concurrently decreased. These results demonstrate that AP2A1 is not merely a marker, but an active regulator that can drive cells towards senescence, or, if its levels are reduced, promote their rejuvenation. Mechanism of action: focal adhesions and integrin transport. To explain how AP2A1 influences cell size, the study examined focal adhesions, FAs, the structures that anchor the cell to the extracellular matrix. Senescent cells displayed significantly larger and more mature focal adhesions, which contribute to a stronger anchorage. The researchers hypothesized that AP2A1 is involved in transporting essential components for the assembly of these adhesions. Since AP2A1 is an adapter protein for endocytosis, they analyzed its relationship with integrin V1, a crucial receptor protein for FAs. It was observed that AP2A1 and integrin V1 co-localize and move together along stress fibers. The proposed model, illustrated in Figure 5e of the study, is that AP2A1 facilitates the endocytosis of integrin V1 and its subsequent transport along stress fibers, which act as tracks toward growing focal adhesions. This direct transport mechanism would be far more efficient in a large senescent cell than simple random diffusion, thus ensuring the necessary material is supplied to sustain the enlarged structure. Conclusions and future implications. This study unveils a fundamental mechanism by which senescent cells maintain their large size. The reinforcement of focal adhesions mediated by AP2A1-dependent transport of integrin V1 along stress fibers. The upregulation of AP2A1 was confirmed not only in replicative senescence, but also in models induced by UV radiation or drugs, and even in epithelial cells, suggesting it is a robust and versatile senescence marker. These findings open up new perspectives. AP2A1 emerges as a potential biomarker for diagnosing the accumulation of senescent cells and as a promising therapeutic target. Modulating its function could represent a novel strategy for developing senolytic or xenomorphic therapies aimed at combating age-related pathologies by promoting cellular rejuvenation.